All right, we're live. Hey everyone. This is Acacia Young. I'm a registered dietitian with Microbiome Labs. And today with me, I have your very favorite microbiologist, Karan Krishnan. And today we're going to talk about some gut infections. So, Karan has prepared a very interesting presentation for you guys. And we're going to have questions at the end. But while we're here, who's here? If you're here, type into the chat box and let us know where you're from, how you're doing tonight. We really appreciate you guys taking time out of your day. I know it's probably late, especially for you guys on the East Coast. So we're really happy to have you here. We got Kristen and Karen, Amy in Rhode Island. Cool. Boise, California. Awesome. Y'all are here from all over.
I think I saw Anchorage as well.
Yeah. Dang. So many people. It's so great to have you all here. I wish we could see all your pretty faces. Cool. All right, so I think we've got about 200 people here right now. Got some Arizona, some Florida, some Dallas. Cool. I don't know how the weather is in Chicago, but it poured in Austin today. And it was like
Good. Wow.
30
Okay.
degrees, which is like a
Oh,
cold for us in August.
yeah. I was in Arizona earlier today, and then I'm in Chicago now.
Oh, yeah?
Yeah, both places are pretty nice. So Arizona is actually, they call it humid. It was like they're like it's so humid, and my nose is all crusted up from being
Yeah,
dry.
yeah.
But I left early, so it was only like 100 degrees at the time. But it's beautiful here, and but with the seasonal changes, it's a perfect time for developing certain infections. So we'll talk about how
Yeah,
some of these things come about.
prime time. Every time I go to Arizona, my skin like starts flaking off. I have to wear lotion.
Right. Same here. Same
Uh,
here.
all right. Oh, see, Dr. Campbell is here. Judy, Sally. Cool. All right. Well, do you want to get started or you want to let?
Yeah, let's jump in. Let's jump in.
All
We'll
right.
get
Cool.
going. I'm warmed up and ready to go. So, thank you guys all for coming. So, many of you who have seen other webinars before know that I do these quite often, but this is the first one I'm actually doing with Microbiome Labs itself. So, this is the inaugural webinar with Microbiome Labs, and we're going to be doing more of these. We're going to be hitting very specific topics. The presentations themselves are not going to be long because the whole idea is to really delve deeper into one particular subject, and then at the end of it, kind of leave you with practical solutions that you can start utilizing tomorrow, if you wish, and kind of give you a different perspective than what you've typically looked at with regards to these conditions. Like today, we're going to be talking about infections and overgrowths, and those are the most common things that you guys are dealing with probably on a regular basis because you're typically dealing with things like SIBO and maybe recurrent C. diff or chronic rhinosinusitis, and all these things tend to be infections. But I want to also kind of paint a bigger picture on what an infection, both a chronic and acute infection, would be, and all of the manifestations that can come from that, things that you may not even think of, and also a different perspective on how you treat some of these infections, because we don't always need to treat infections by going after the infective agent. In many cases, that can actually do more harm than good. And so we'll talk about the ecology of the microbiome a little bit as well. So it's just enough to give you some good and practical information, and then we'll open it up to questions and all that as well. And Acacia here will facilitate all of that, and I'm sure she'll jump in with thoughts whenever she wants. But let's jump in and start the screen sharing. So I'm going to share my screen, and we'll go right to a PowerPoint that you guys can see.
So let us know if you guys can see it because it's going to adjust for a second.
Okay, Acacia, are we all good? Does it look good?
Looks good from my end. Can you guys see everything? If you can, just type in the little text box and let us know that you can see the slideshow.
Okay, so let's jump in. Infections and overgrowths. the first thing to note, and I've said this many times, is we really have to start thinking of our bodies as a superorganism, a holobiome. I've used that word before, and it's probably one of my favorite words. It's interesting to say at cocktail parties because it makes you sound either really smart or nerdy. So you'll either make friends or people will not want to hang out with you. But either way, it's a really important thing. Holobiome is a superorganism. Our body is really made up of this very, very complex ecology, and any perturbation in that ecology has huge downstream effects. You'll see in some of the upcoming slides, I try to illustrate how this can happen in a sequential way, so that it's easier to kind of look back when you're dealing with your patients, playing detective a little bit to try to figure out what the cause of their actual condition could be. And a lot of that will come from taking a really detailed and effective history to really understand, the experiences they've had that could have led to the condition that they're dealing with. So we'll start the presentation by defining a number of terms, which I think are important to clarify. Many of these terms you guys are very familiar with, and of course know the definitions well of, but I thought it's important to kind of lay that groundwork, and then we'll jump into the meat of the subject itself. But the first thing is to define diseases versus infectious diseases. So a disease really is any deviation from a condition of good health and well-being. That's pretty self-explanatory and known, but an infectious disease is a disease condition caused by the presence or growth of infectious microorganisms or parasites. Now, what's important here is that as we come to understand the impact of the microbiome on overall health, we start to understand that most diseases that we know are really are caused by infectious diseases. So the bigger picture here is that a dysbiotic gut is not just an imbalance in your flora, it's actually a chronic infection. So we'll elaborate on that a little bit more. So for example, heart disease. Heart disease is still the number one killer in the United States, and there's so many interventions for heart disease. Of course, statin drugs and surgeries, and the list goes on and on. And yet the prevalence rate of it keeps increasing. It's because we're not looking at heart disease as a downstream effect of a chronic infection. So that's something to keep in mind. We'll elaborate on that as we go along. But so I think this distinction is really important because when your patients present with a disease, more often than not, you could trace it back to a chronic infectious disease that is the true cause of their ailment. The next thing is to define pathogenicity versus virulence. This is an important distinction in my mind because it gives you an understanding of the different classes of microbes. Pathogenicity is basically the ability of a microbe to cause a disease. So some microbes within your system cannot cause disease because they don't have the ability to do that, meaning they don't produce toxins, they don't produce virulence factors, they just don't have it in their genetics to cause disease. Many of them do. And this term is often used to describe and compare species of bacteria. So some are pathogenic bacteria like Clostridium difficile, Clostridium perfringens is not necessarily pathogenic. Virulence is really the degree of pathogenic. So you have organisms that are really pathogenic. Look at Ebola, and look at the plague, for example. These are heavily pathogenic and very strong virulence organisms versus something like the common cold, which has a very low virulence factor. A lot of people can be exposed to the common cold and actually not end up with any sort of symptoms at all. And that's the same with many foodborne pathogens and pathogens we come in contact on a daily basis on surfaces that we touch and so on. We're exposed to them on a continuous basis, but because their virulence factors are low, their ability and their degree of potency in causing disease tends to be low as well. And that's an important distinction because the vast majority of microbes that we come across on a daily basis have very low virulence and very low pathogenicity. They're typically not really harmful to us, and in many cases, they're beneficial as well. This term is often used to describe strains within a different species as well. So if you take certain types of influenza viruses, for example, H1N1 has very high virulence versus your regular wild type influenza. that we tend to come in contact with quite frequently. So that tells you the degree of potency of their ability to cause disease. Acute infection versus chronic infection, most of you know what this is. Acute infection is one that's characterized by sudden onset, rapid progression, often severe symptoms. This is the one that gets the most attention when people are talking about microbial or viral or parasitic associated infections. But really, the one that plagues us are the chronic infections, because these are infections that are characterized by delayed onset and slow progression. And in fact, we may not even see the manifestations of these infections for years, maybe even decades. Just look at, for example, acquired immune deficiency syndrome, HIV. You might get infected with HIV and never see symptoms for 10 years with regards to HIV. So there's many different ways of looking at this, but one thing is that chronic infections tend to be organisms that either have non-obvious symptoms or symptoms that take a long time to show up. So they're causing disease in your body, but you're not really noticing the symptoms yet. And so that's within chronic infections, typically you have a primary infection versus a secondary infection. And so things typically start off with the primary infection being acute. And the primary infection is usually an infection that develops in an otherwise healthy individual. That could be a cold, a flu, a sinus infection, a UTI, something that causes that first insult. And then a secondary infection tends to follow that, and that's an infection that develops in an individual who's already infected with a pathogen. And what we tend to see is that those chronic infections I talked about that cause the diseases that we deal with on a regular basis are typically secondary infections to a primary insult. I'll give you an example of that as we go along. Localized infection versus systemic. Of course, localized is an infection restricted to a specific location, and systemic is one that is spread throughout several regions of the body. And now this could be with the presence of the microbe or without. We'll talk about the difference there between toxins and how microbes can actually infect within the body itself. Clinical infections versus subclinical infections. And this is really important because this kind of delves into the chronic infection that I'd mentioned earlier, because a clinical infection is one with obvious and observable detectable symptoms. So that's like, you get sinus infections and you see all of the symptoms associated with it right away. And you can measure those, and then the symptoms tend to go away once the infection clears. But subclinical infections are infections with few or no obvious symptoms, but they're still infections. And that's really what I was talking about earlier, where the vast majority of diseases we deal with are a consequence of chronic subclinical infections going on in the body. And that can be as simple as a dysbiosis. A slight imbalance in an optimal flora is actually a subclinical chronic infection, because it's driving a disease progression that's not obvious at the moment, but down the road has significant symptomologies. And I'll show you an example of how that can be. Opportunistic infections are any infections caused by microorganisms that are commonly found in the host environment. So they're typically not organisms that come in from the outside, but typically reside within the microbiome itself. And this term is often used to refer to infectious diseases by organisms within the normal flora itself. Think of things like C. diff, certain types of Enterobacter, E. coli, even viruses that remain dormant within the body, EBV, cytomegalovirus, these things that can remain dormant for a while. And then because of perturbations in the environment, they can actually change. So they tend to capitalize upon ecosystem alterations. And when they see the opportunity, they go ahead and they start the infective process. Here's an example of how a perturbation in the environment can allow an opportunistic infector to proliferate, but then also show you how a primary infection can lead to a secondary and even a tertiary infection and start the process of chronic disease. So this is an example of that. This is the impact of antibiotics on the microbiota, and then expansion of what we call enteric pathogens. So imagine that an individual had, let's say, UTI or a sinus infection, and they took a course of antibiotics. Here in the first part in the section labeled A, we've got a normal, healthy, diverse microbiome, a dual layer mucosa. The bottom part of the mucosa is really thick, which makes it more sterile so that microbes and toxins and things aren't leaking through. And you have a healthy intestinal epithelial layer as well. Now, let's say you take a course of antibiotics, and what happens is the antibiotics reduces the diversity of the microbiome. And in that reduced diversity of the microbiome, Clostridium difficile, which is a normal part of the commensal flora, is an opportunistic infector and can now start to proliferate in the presence of low diversity and start to produce its toxins. So this is the secondary infection that is opportunistic. The primary infection is the reason we're taking the antibiotic in the first place, whether it's a UTI, ear infection, or sinus infection. Now it starts to produce its toxins enough, and these toxins actually can migrate through the thicker layer of mucin and damage the intestinal lining and starts the process of colitis and cell death. So now you're starting to see how a secondary opportunistic infection can actually start a chronic disease like colitis. Now, here's the thing that can happen that can actually lead to more chronic infections in the same scenario and can actually lead to further gastritis and problems. So antibiotics can also then increase the amount of something called free sialic acid from the host. So the host produces sialic acid in response to the presence of the antibiotics and lowering of the microbial diversity. And then you can also get certain microbes within the gut, commensal bacteria like Bacteroidetes, that'll produce higher levels of succinate from the microbiome when antibiotics are present. So what happens is, as a bacterial diversity lowers because of the antibiotics, Bacteroides produces succinate from digesting certain types of carbohydrates within the body. Not exactly sure why they produce higher levels of succinate in lower diversity microbiomes, but they tend to do that. Now, the elevation in succinate and salicylic acid can actually activate Salmonella typhimurium, which is another microbe that could be part of the microbiome, but not really infective until things like sialic acid and succinate are at higher levels. And so then Salmonella typhimurium starts to produce its own toxin, penetrate through the thick layer of the mucosa and start causing gastroenteritis. So now what you have is the elevated succinate and salicylic acid also allows C. diff to continue to proliferate, which can cause further colitis at the same time as having gastroenteritis. So now what you have is this chronic disease that's eating away at your intestinal lining and causing long-term issues, and of course numerous spiraling issues from there that all started from a single dose of antibiotics for a sinus infection or UTI, something like that. So the point here is that this cascade is happening quite often, but it doesn't necessarily have to happen in short term, meaning somebody could be suffering from colitis in their mid to late 20s, and it could be a progression from a course of antibiotics they took in their early teens. This whole progressive dysbiosis, secondary opportunistic infections, tertiary infections, and then the subsequent chronic illnesses that come about can take a decade to show up, right? So, and the presence of this imbalance within the gut, the presence of the increased salmonella, increased clostridium, will continue to perpetuate the illnesses. It won't allow for the intestinal epithelial to heal and all that. So the idea here is that if someone shows up with a gastroenteritis, or they show up with colitis, more than likely the disease was set off in motion, a year, 6 months ago, or as far as 10 years ago. And the primary thing to do is to address the dysbiotic microbiota that is allowing the perpetuation of the disease. So the secondary and opportunistic infections are probably still there, driving the illness. Here's another illustration of this, and this is the modulation of enterohemorrhagic E. coli. virulence factors. So enterohemorrhagic E. coli is a type of E. coli that's found in the normal commensal flora. Again, it's an opportunistic grower. It is pathogenic. It has medium virulence. It's not super virulent. So it needs the right environment in order to become pathogenic. Now, this is non-antibiotic perturbation. So I didn't want to just show antibiotics that can actually hurt the system and cause this cascade, but there's many non-antibiotic ways of causing perturbations that lead to this type of infection as well. So for example, any reduction in motility due to things like dietary changes, travel, medications are probably one of the most common reasons for reduction in gut motility. It can actually increase the production of succinate by commensal bacteria called Bacteroides theta-iodomicron, and this organic acid actually helps this type of E. coli, this enterohemorrhagic E. coli, and even C. diff to become infective. So again, this is your common commensal good bacteria, this Bacteroides theta-iodomicron, but because of the reduction in motility, so now your peristaltic activity and the cleansing and movement of the bowels has slowed down quite a bit, and in response to that slowing down, your commensal bacteria is producing more of these organic acids that will actually increase the infectivity of commensal opportunistic bacteria. So in this case, now you've got enterohemorrhagic E. coli. Once it starts being allowed to grow at higher rates, it produces things like mucinase, which is an enzyme that breaks down the mucin protective layer, and that allows the E. coli to actually bind to the intestinal epithelial cells and cause infection and changes in colitis and so on. So this is a non-antibiotic perturbation. Things like as simple as changing your diet drastically or traveling overseas or medications can lead to this secondary infection that starts to create disease. Now, conversely, if Bacteroides theta-iodomicron digests small parts of the mucosa and release fucose, it prevents the virulence of EHEC. So this is really interesting because what we're talking about here is, let's say you have a perturbation in the motility of the gut, right? And that comes from medication that you just started taking. In the next slide, I'll show you several medications that can actually lead to disruption in gut motility that can lead to this whole cascade. But let's say that you take the medication, it disrupts motility, you start to get more succinate production from Bacteroides, and then now you're starting to get more prevalence of E. coli. If the individual was fasting, for example, the Bacteroides theta-iodomicron would in fact digest small parts of the mucosa instead of incoming carbohydrates, and instead of producing succinate, it produces fucose. And when it produces fucose actually acts as a prevention for the virulence of enterohemorrhagic E. coli. So the person that's taking the medication that's creating a reduction of motility, but they're fasting, they happen to be fasting as well, will never see this progression into developing disease. But the person who's not doing the fasting will actually see the production of succinate and then the virulence of E. coli, and then, of course, the subsequent infection of the intestinal epithelial cells. So just that simple difference will actually lead to the disease or not lead to the disease, even in the course of perturbations. So here's some medications that actually cause perturbations in the motility of the bowel movements. These things like Benadryl, anafranil, thorazine. These are all very common medications that are typically used for allergic symptoms, and these actually dramatically reduce the motility. Tricyclic antidepressants also do that. Of course, antidepressants are very widely used, so it's not at all uncommon to see somebody coming into your clinic who's been on antidepressants or who are on antidepressants, and because of that, they actually have the colitis. You would almost never connect the antidepressant to the colitis without really going through this progression of disease, because the antidepressant would have slowed down motility. The slowing down in motility can create changes in metabolites being produced by the microbiome. The changes in metabolites lead to opportunistic and secondary infections. The opportunistic and secondary infections lead to the disruption in the mucosa and then the infection of the intestinal epithelial cells. So that's a progression. All starts with just starting one of these medications, especially one of these anti-allergy medications as well. So beta adrenergic agonists like Proventil, for example, that people use for asthma all the time. It's which is a pandemic going on. So between allergies and asthma, you've got many of these medications covered, and these same people are the ones that end up with all of the gut infections, SIBO, overgrowth of Candida, and so on. Of course, opioids, as we know, there's an opioid epidemic going on in our country. Oxycontin and Vicodin and all of these things, Demerol, are very potent anti-motility agents. So a lot of common medications can lead to the non-antibiotic perturbation of the gut, which leads to secondary and tertiary chronic infections. Even things like chronic mineral imbalances can lead to poor motility. So people who tend to be undernourished, don't get enough minerals. Minerals are really important for the electrical activity that causes peristaltic activity and the migrating motor complex to improve gut motility. So even something as simple as just having mineral imbalances can lead to colitis and gastroenteritis, or even IBD, IBS, and so on. So it's important to keep in mind this pattern of progression of disease, because then when you look back to figure out what are the things I can do to for the patient to improve their condition, you start to be able to look back at the true cause of things. In many cases, it's as simple as motility issues. So the normal flora of humans tends to have numerous types of organisms, which we can be categorized typically as symbionts or symbiosis, but there are many different types of symbiosis, and that's one of the things that most people don't really pay attention to. The word symbiosis in general tends to have a positive implication, and people typically think symbionts are mutually beneficial, but that's not true. There's many different types of symbiosis. For example, mutualism is the best kind. It's a symbiotic relationship in which both species actually benefit. But commensalism, which is the vast majority of bacteria within our gut, are symbiotic relationships in which one species benefits and the other species is neither help nor harm. So a lot of the microbes within our gut are just kind of coming along for the ride. Now, they may have some downstream benefits that we don't really know about yet, but the vast majority of species within our gut are commensals that just kind of act as part of the flora, and we're not really sure, we can't pinpoint exactly what their specific benefit is to us. Now, the other thing is parasitism, which is actually another type of symbiosis, which is where it's a symbiotic relationship and where one species benefits and the other species is actually harmed. But again, a lot of parasites do have certain benefits as well. A lot of worms and helminths and things like that tend to improve allergic symptoms and improve immune function. So we're starting to see a lot of overlap between symbionts, commensal organisms, parasites. We're starting to understand that the whole ecology seems to be important, and it's becoming harder to kind of pinpoint each organism and figure out whether they're good or bad or indifferent. And generally, the species that benefits the parasite tends to be much smaller than human, which is the big host. Virulence factors and toxins, just to give you a little bit more info on how these microbes tend to infect, there are numerous enzymatic virulence factors, things like coagulase, which is produced by Staph aureus, streptokinase, hyaluronidase, which many pathogens produce, collagenase, leukocidin, and hemolysin. Hemolysin is one that's produced by a lot of E. coli species that are infective. They basically break up red blood cells and cause bleeding and things like that. Other ones are adhesion factors, things like protein A, protein M, both by staph and strep. And then the ones that are most common that we deal with are exotoxins, which is a type of bacterial toxins that tend to be produced by gram-positive or gram-negative bacteria, but are excreted by the bacteria. So it's not embedded within the bacteria itself, it's excreted. The action of exotoxin actually doesn't require the presence of the bacteria. It could have been excreted into a different environment that you end up picking up the toxin without picking up the bacteria. I'll give you an example of this. When I first started in the food microbiology side, one of the projects I worked on is egg whites. So one of the things that they do with egg whites, of course, is they collect egg whites and then they reduce something called the Maillard reaction, which is a browning reaction within egg whites. So there's this process of treating egg whites with certain enzymes to stop the browning reaction. And then once the browning reaction is stopped, then they can ship off the egg whites to for use in producing eggs, producing both scrambled eggs, liquid egg whites, or going off to make Twinkies or whatever it may be. But one of the interesting things about egg whites is they tend to have a very high concentration of exotoxins because salmonella, for example, that is quite prevalent among chickens and birds. You also find it on the eggs themselves when the birds lay the eggs. So then, when the machines are cracking the eggs to extract and concentrate the egg whites, the cracking action actually spreads the salmonella exotoxin. And that salmonella exotoxin ends up being found in the egg white itself. And so you don't have the bacteria there because they'll sterilize the egg whites before use, and so the bacteria will more than likely be dead, but the exotoxin is actually still there and can still make you sick. So that was an interesting application. So our whole focus was how to reduce the presence of this exotoxin in the egg whites that were being shipped out to different places. And a lot of them tend to be heat sensitive, but some of them are not. For example, Staph aureus produces an exotoxin that's very heat stable, and so even the cooking process can't necessarily kill it. So you might get sick short term from eating something without any bacteria being present in it. It's because of the exotoxin the bacteria produce at some point. Other ones, classes of exotoxin can be neurotoxic, cytotoxic or neurotoxic, interferes with synaptic transmission, so it messes up your neurons. Cytotoxic is anything that inhibits cellular activity, whether the cells can metabolize or absorb nutrients, and so on. It can disrupt how the cells function. Enterotoxins interfere with water absorption in the large intestine and basically irritates the lining of the gastrointestinal tract, causes things like diarrhea, constipation, and so on. Other virulence factors, and this is the most prolific virulence factor, is endotoxins. Endotoxins are a type of toxins that are basically produced by just gram-negative bacteria. They are components of the gram-negative bacteria cell wall, so they're a normal part of the cell structure of the gram-negative bacteria. And the action of the endotoxin requires the presence of the bacteria itself because the toxin is actually embedded within the living bacteria. And then the endotoxin gets released when the bacteria dies, typically, or the cell wall disintegrates. And so then the breaking up of the cell wall is what releases it. And the endotoxin is a normal part of how the bacteria communicates with one another. They use it as a receptor in many cases, but it happens to be a very, very potent toxin. Now, keep in mind that the vast majority of bacteria in your microbiome are gram-negative bacteria. So the vast majority of your commensal bacteria carry endotoxins in it. And the endotoxins is something called LPS, which has a lipid A component. And the mode of action, and the lipid A, sorry, let me elaborate on that. Lipid A is part of the lipopolysaccharide layer. The lipid A is a component of the endotoxin that's toxic. It's got something else called the O antigen and so on. That part's not that important, but the whole point of it is the endotoxin has this lipid component to it that is highly toxic to human cells and causes, at the low end, irritation and inflammation, but it can also really get into tissues like our amygdala in the brain or hippocampus and penetrate the blood-brain barrier and actually cause inflammation in the blood-brain barrier. It can get into the dorsal root ganglia and stimulate things called nociceptors, which cause the feeling of chronic pain. So these toxins that are components of your own commensal bacteria can be released into your system that cause all of these symptomologies, including systemic and localized inflammation, GI irritation, total disruption of your epithelial cells causing leaky gut, even hemorrhaging blood cells and so on. So it really becomes quite messy. So now I wanted to just kind of go over a couple of slides on acute and chronic infections associated with chronic disease. So this is where we'll follow the path of a of disease progression so that we can see what the end result looks like. And this is the point in which somebody's probably coming into your clinic, but we can kind of trace it back to see where the issues may be to really help them deal with the issue that they're dealing with. So for example, let's follow the destructive path of a single infection. Let's take chronic rhinosinusitis. So you get people that have chronic sinus infections. So what they tend to do is frequent nasal rinses and aggressive nose blowing. And interestingly enough, come to find out, there's studies that have been shown that chronic and frequent nasal rinses can actually make sinusitis worse. So I know people. I used to be one of those people as well several years ago that tended to have chronic issues with my sinuses, and so I was doing one, maybe two nasal rinses a day. And of course, when you're done with the nasal rinse, you have to blow your nose quite aggressively. And studies are showing that aggressive nose blowing actually blows back infected sputum and infected mucus lining deeper into your sinus cavities. And so it actually increases your risk for a sinus infection. And so somebody that's going through this chronic rhinosinusitis and maybe doing these lifestyle things, frequent nasal rinses, aggressive nose blowing, and then they end up with an actual sinus infection. And for that sinus infection, they have to take a course of antibiotics. And many times they take multiple courses of antibiotics to deal with the infection. But let's just talk about one course of antibiotics. And that one course of antibiotics will start off the process of GI distress. Like we talked about, because the presence of antibiotics causes bacteroides within the gut to produce more succinate, salicylic acid, and so on, it starts the progression of dysbiosis. Now, that person 6, 8, 10 months later might get another sinus infection because they are still continuously doing nasal rinses and blowing their nose hard. The second or third course of antibiotic will cause severe disruptions in the diversity, which can then lead to chronic opportunistic infections, and then even things like stasis, which is the loss of motility within the gut. Now you have a real defined dysbiotic gut that's going to lead to chronic opportunistic or even non-opportunistic infections. Now, this chronic infection causes the development of SIBO because now you've got E. coli and other enterics that are starting to grow at higher rates than they should in the small bowel because the motility has been disrupted by the use of the antibiotics and the disruption of the microbiome itself. And so the concentration of these unfavorable microbes starts to increase really high. Now this person is starting to show symptoms of SIBO. Now with the SIBO, it leads to things like gluten intolerance because now the oral tolerance that you're supposed to be developing is getting disrupted because there's chronic immune activation in the small bowel and in the mucosa lining itself. So the SIBO leads to perturbations in the immune function, leading to gluten intolerance. So now this person not only has symptoms of SIBO, but they're becoming much more intolerant to gluten. But it's their birthday, and they go out one night and they have some birthday cake and they have a few drinks that contain gluten. And that is enough of a trigger, an environmental trigger, to end up causing chronic skin disorders. So things like atopic dermatitis or psoriasis or even, eczema on the face or cystic acne that they now are dealing with. So now at this point, they're coming into your clinic with this condition that just cannot be treated with medication anymore. And they want you to figure out what is going on. And all of this could have started with their sinus issues and the frequent nasal rinses and aggressive nose blowing. So it just wanted to show how things can progress to lead to this middle step of developing chronic GI distress, dysbiosis, and loss of function in the GI, things like motility, that can lead to sensitivities and then express itself as an autoimmune condition. Now, this person could have ended up with Hashimoto's, they could have ended up with lupus, they could have ended up with rheumatoid arthritis, many, many different things, but it all starts with this kind of progression. So I just thought it was an interesting way of kind of deconstructing where the issue may be coming from, because when this person walks into your office, you may not be thinking about their sinus issues, being the trigger of this whole cascade, and then their practices for the sinus issues as well. So once you start getting into that mindset of investigating the perturbations and where the chronic infection is coming from, that might lead you to understand their lifestyle choices that could be leading to those perturbations as well. So same thing with any loss in motility, which leads to SIBO, can create infections in the gut that are chronic infections, but it can also create chronic diseases. So the immediate loss of motility for whatever reason, it could be the use of antibiotics, non-antibiotic perturbations, medications, all of those things can lead to further chronic infections and then chronic disease as well. So what can we do about all of this? There are four really important tasks to reduce the risk of chronic infections that lead to chronic disease. So these are things just to keep in mind to either be utilizing with patients that are already showing symptoms of severe chronic disease to try to help the progression, slow down the progression of disease, or even reverse it. But it can also be used as prophylactic for prevention of disease in case of perturbations. Remember I gave you that example where that person who would have had that loss in motility and increased production of succinate by Bacteroides would have and increased infection by E. coli. That would have been prevented if that person was fasting. And so these are things that you can do on a regular basis to help your body deal with perturbations that may occur from time to time so that it's not as sensitive, so you can kind of be a lot more resilient against them. So number one is to improve and maintain microbiome diversity. We'll talk about each of these more specifically. Improve the mucosa and gut motility. So gut motility is such an important thing. We really don't think about it enough until people are constipated or people have SIBO. But even well before those symptoms, people could be having reduced gut motility that can be setting up the stage for chronic infection. Increases in secretory IgA becomes extremely important. There are numerous things that can lead to selective IgA deficiency. Selective IgA deficiency is just deficiency in IgA specifically and no other immunoglobulins. Selective IgA deficiency is now being shown to be one of the kind of foundations for many chronic illnesses because IgA is our first line of defense. It's the immunoglobulin that's produced at the highest levels within the immune system. We produce upwards of 7.5 to 10 grams of it a day, which is a huge amount when you're looking at things that are tiny, molecular structures. And so IgA becomes extremely important in neutralizing overgrowths and infections, and that's a very important line of defense. And of course, increasing things like Treg, which is T regulatory cells that suppress unfavorable immune responses, and then also improving overall immune function in general. So let's look at each of these in a little bit more detail. Number one is improving and maintaining microbiome diversity, and that comes from increasing the diversity of your diet. The more different foods we eat, the more different our microbiome becomes and the more diverse it becomes. For example, if you look at hunter-gatherer tribes like the Hadza tribe in Tanzania or the tribes in Papua New Guinea, when they study them and they've studied their diet on a very intimate basis, actually by actually living within these tribes and eating the way they do, they consume anywhere between 400 to 600 different types of foods on an annual basis. 400 to 600 different types. And anthropological evidence shows that early humans, our ancestors, consumed about the same amount, around 400 to 600 different types of foods. In the Western world, we consume around 15 to 20 different types of foods on a regular basis, and that has a huge impact on the diversity of our microbiome. We know that a diverse microbiome is resilient against opportunistic or any other kind of infection, whether acute or chronic. So a diverse microbiome is resilient to changes that you might be going through on a daily basis, whether you've traveled somewhere, you ate something you shouldn't, or even a single course of antibiotics. Your microbiome may bounce back better if it's more and more diverse rather than being less diverse. So more complex carbohydrates from seeds, roots, and tubers. Even people with SIBO can handle these better These tend to be low FODMAP things, so seeds, roots, and tubers are extremely important. And then just eating a little bit of a lot of different things. Utilize spores, of course, like megaspore for competitive exclusion to get rid of the infected microbes that are trying to overgrow and also help recondition the gut itself, so we can make significant changes just utilizing the spores itself. And we see that quite frequently with patients. And then, of course, intermittent fasting. As I mentioned that earlier, intermittent fasting actually has a significant number of benefits. One of those is actually the increase in diversity of the microbiome. There are certain species that proliferate really well during fasting periods, and they don't do very well during fed periods. So the only way to get those species to come up and start increasing the numbers is to allow your body to go through certain intermittent fasting periods. That's typically a 16-hour fasting period is fairly normal to get those kind of benefits. Now, how do you improve the mucosa and gut motility? One is the increase of these 4 amino acids. There's been 1 or 2 studies that have shown that these particular 4 amino acids are very important in the construction of the mucus layer. Now, remember, the mucus layer is a very important barrier because the top part of the mucus layer tends to be fairly liquidy, and most of the microbes that live within your microbiome exist in the top part of the mucus layer. The bottom part of the mucus layer is made up of something called mucin 2. It tends to be very thick, very complex, and sterile. The whole part of the bottom part of the mucus layer is to create a barrier between all of the stuff that's going on in the digestive system and the lumen of the intestines, in the microbiome, and the intestinal epithelial cells. So your body has this really important barrier. Now, that barrier is made up of glycoproteins. And the 4 main amino acids that construct the glycoprotein are threonine, serine, proline, and cysteine. Now, L versions of these are extremely important. So you want L-serine, L-threonine, L-proline, and L-cysteine. These amino acids alone can help improve the production of mucus, and especially that dense, thick layer of mucin 2. Increase the intake of insoluble dietary fibers, of course, assuming some of these don't irritate some of the SIBO people, but many of these should not irritate SIBO people as much, because they tend to have much more complex carbohydrates, and they're insoluble as well, so they'll get down into the lower part of the intestines much better than other forms of soluble fiber. Fasting, of course, as mentioned before, fasting really increases gut motility, because when you fast, you actually stimulate something called a migrating motor complex. That's an electrical wave that moves through your intestinal lining. It clears out your stomach, the distal part of your stomach. It clears out your proximal small bowel, your distal small bowel, and all the way moves things into the colon. And fasting even in between meals becomes important, because if you eat a meal, then the migrating motor complex stops. And if you eat again too frequently from that meal, you never allow the migrating motor complex to kick in after that meal in order to start pushing all the stuff that you consume down so it doesn't accumulate in the small bowel. So not only is it important to have a long period of fasting throughout the day, meaning overnight and then through the morning having that 16 hours, but there's also an importance in fasting in between meals, meaning giving yourself adequate time in between meals, typically around 3 to 6 hours is normal. And so if you're eating every 2 or 3 hours, especially if you have SIBO or overgrowth, that can actually perpetuate the issues, because it's reducing motility. Increasing butyrate. Butyrate is one of the most important things to increasing the production of mucosa. There are cells within your intestinal epithelial cells called goblet cells. Those are the main cells that produce mucin. They are stimulated by butyrate. Butyrate, of course, is primarily produced by the bacteria within your gut itself and within your large intestine. And what they feed off of to produce butyrate are things like resistant starches. Now, if people can't handle resistant starches because of the SIBO, then you can also use things like chondroitin sulfate or hyaluronic acid. Most people don't know that chondroitin sulfate and hyaluronic acid can be utilized by bifidobacterium species, for example, to produce butyrate and other short-chain fatty acids. You would never think of chondroitin sulfate and hyaluronic acid as a prebiotic, but it actually is. So this is a little tip for those. So if you have SIBO patients who can't deal with any sort of resistant starch, any sort of oligosaccharides like FOS or GOS or XOS. Try some chondroitin sulfate with them. Try some hyaluronic acid. These are things people typically take for joint pain, but you can actually use them as prebiotics as well. Now, of course, spores increase the production of butyrate by about 40% compared to normal healthy flora. So, of course, taking your megaspore will have a huge effect there as well. Now, prokinetics become really important, things that stimulate the motility, things like coffee, ginger root, garlic, magnesium, even exercise has a very good stimulatory effect on the kinetics of the bowel, so we get the bowels moving again. Because remember, the gut motility is a really important thing at preventing the onset of chronic and secondary infections. And parasympathetic activation. This becomes really important because a lot of people who tend to have SIBO, fungal overgrowths within their system, secondary and opportunistic infections that have led to things like colitis, IBD, and all that tend to be sympathetic dominant and don't have activated parasympathetic systems. I won't go into all different ways of activating parasympathetic system, but it can be as simple as humming in the morning, doing deep diaphragmatic breathing, splashing cold water on your face. If you just look it up, there's many different tactics of activating parasympathetic system. One of the things that we're seeing is high doses of vitamin K2 actually changes sympathovagal tone and actually upregulates parasympathetic system as well. So that may be another little trick. You may not think to treat your SIBO patient with vitamin K2, but vitamin K27 can upregulate the parasympathetic activation based on some of the initial studies we're doing right now. Now, the next thing is, how do you increase secretory IgA? Well, number one, low stress. So stress actually is an inhibitor to secretory IgA production. There's a number of GPRs, which are receptors within the intestinal epithelial cells that need to be stimulated in order for your body to produce secretory IgA. Stress hormones are inhibitors to this cascade. So it becomes really important for people who are dealing with chronic Candida, sinusitis, chronic UTIs, or even SIBO, to start looking at mindfulness practices, yoga, and even massage. So massage, to give you an example, increases something called oxytocin. Oil massages can increase oxytocin levels by as much as 30%. And when oxytocin levels go up, stress hormone levels go way down. And so there's been studies that have been published to show that even a 1 hour oil massage can actually upregulate oxytocin levels in a very significant way and reduce corticosteroids and other stress hormones by quite a significant margin as well. So, you would never think to recommend to your SIBO patient or your Candida patient, go for a massage once a week or maybe even twice a week. We're talking about just an hour massage or 50-minute massage with oil can increase the oxytocin to actually improve secretory IgA production. Increase moderate exercise. Exercise seems to be something that induces secretory IgA. Intake of these particular nutrients, in particular choline, vitamin C, zinc, colostrum does a great job of increasing secretory IgA. Beta glucans seem to be converted into short-chain fatty acids, but also can be utilized immediately by your immune system to produce secretory IgA. Proteins, omega-3 fatty acids, all seem to have an impact on increasing secretory IgA. And then, of course, more exposure to environmental microbes, just getting out there and getting dirty a little bit more, because environmental microbes will increase your body's alertness and increase your body's production of secretory IgA. And then increasing Treg and overall immune function. Of course, the spores do this amazingly well. Many of you know that already. Increasing butyrate by resistant starches and other techniques we talked about in previous slides dramatically increases the function of Treg in your overall immune system. Vitamin A seems to be really important in Treg function, anywhere between 10,000 and 15,000 IUs a day. In fact, a mixed carotenoid is probably a better way of getting some vitamin A. Beta carotene is probably the better form of vitamin A, but taking mixed carotenoid is a good way. DHA seems to play a role in increasing Treg function. Curcumin, cumin seed oil, and of course sleep. Sleep is extremely important for overall immune modulation and increasing Treg function as well. So that's the overall thing. I'll leave it on this slide just to give you a little conclusion. So the whole point here is to be able to think about diseases that your patients are presenting to you in terms of perturbations in this holobiome, right? We are, as I mentioned earlier, a walking, talking rainforest. We are a very complex ecology. And so minor perturbations in that ecology could be chronic nasal rinses. It could be a single course of antibiotics that were taken 5 years ago for something can lead to this cascade, which is presenting itself as rheumatoid arthritis or presenting itself as eczema, psoriasis, all of these things, or anxiety, depression, all of these things can be traced back to some form of chronic subclinical secondary infection. And the best way to treat those things is to upregulate your body's natural defenses against these things, including the microbiome. You'll notice in here, I didn't make any recommendations on antimicrobials, right? Which many people use when they suspect SIBO, they suspect, overgrowths of E. coli or enterics, or even you do a stool test and you see high levels of enterics, and you might hit them with antimicrobials quite a bit. The problem with that, and antimicrobials can be effective and they can be utilized, but the problem is we don't necessarily know what kind of organisms are causing the problem and if those organisms are going to respond to the antimicrobial, or if the antimicrobial is actually going to perturb the ecology even more and cause more secondary or tertiary infections. So that's why I'm always very cautious with antimicrobials. And my recommendation from a microbiology standpoint is when someone presents with something that is likely caused by a chronic infection, the first steps should be to try to restore the microbiome and restore the mucosa and restore your secretory IgA in order for your body's own defensive mechanisms to take care of those infections. Because remember, most of those infections are low virulence infections. They're not very virulent organisms. Just a small shift, a small increase in the good bacteria can shut them down. A small increase in the right type of microbes being introduced in the system can shut them down. So rather than hitting them with antimicrobials first, look at considering ways of improving their current microbiome and improving their defensive systems like the mucosa, the gut motility, and so on, before hitting them with antimicrobials. It's just something to keep in mind. Now, every clinical situation is going to be a little bit different, but my job here is to kind of give you a slightly different clinical perspective on things that you could potentially be doing. So we'll leave it at that, and let me see if I can stop sharing, and maybe we'll go to questions.
Yeah, so we do have a lot of questions for you. A lot of people have questions about Xifaxan in particular.
Yes, okay.
So this one is just, what about Xifaxan as an antimicrobial, and what about antifungals?
Can you see me now?
Yeah.
Okay. Okay. All right. Can you repeat that question? Because it went blank for a second.
Yeah, the question was, what about Xifaxan as an antibiotic? Since a lot of practitioners are going to be using that, since it is so specific, is it still going to cause all the same issues that a broad spectrum antibiotic would cause?
Yeah. So what's interesting about Xifaxan is that according to the company, and I've looked at some of this research, it looks good, but you never know with pharma companies because they often hide studies that don't look good. And so the studies showed that Xifaxan doesn't really enter the large bowel, right? So it's not supposed to cause major disruption in the large bowel. It's supposed to focus its activity in the small bowel, where it can disrupt overgrowths within the small bowel and then be cleared from the gut within about 6 or 7 hours. However, the rebound reaction from Xifaxan is quite present. So, for example, a lot of patients who are severely SIBO or have other conditions like hepatic encephalopathy, where they have overgrowths of prevatellas and things that produce a lot of ammonia, they have to be on Xifaxan for many courses or even chronically in many cases. And what that's telling me is that the targeting of the microbe by just killing it off in one shot with a course of antibiotics is not really resolving the problem, because you probably still have issues with motility, probably still have issues with overproduction of certain metabolites within the small bowel, like succinate or salicylic acid. So we're not getting after the real problem, which is restoring the structure and function of the bowel itself. So even before Xifaxan, my recommendation would be to try some of those other techniques to try to restore the bowel function, restore the mucosa, restore motility, improve prokinetics, improve the migrating motor complex, try to increase microbial diversity. And if those things aren't really helping, then maybe hit it with an antimicrobial like a Xifaxan or herbal antimicrobial, and then go back to those things immediately to try to restore the gut.
Yeah, that ties us into another question, which was, is there a better way to administer antibiotics that can protect biodiversity in this way?
So and that may be the reason that's based on why they're taking the antibiotics. So studies have shown that IV antibiotics are less disruptive to the gut microbiome than oral antibiotics. So oral antibiotics, of course, have to clear through the gut. Average oral antibiotic, like you take a clindamycin, for example, clears it through the gut around 2.5 to 3 hours. And so they tend to have that disruptive effect. So if it's something that it's an infection that is distal to the gut, it's somewhere in the peripheral part of the body, maybe look at IV antibiotic instead, or a shot like we used to do penicillin shots back in the day. That may have less disruption on the microbiome. Now, how do you take an oral antibiotic and reduce the disruption? That's really kind of tough, because it's really hard to control the effect of the antibiotic. The key is going to be to take a right probiotic with the antibiotic so that the disruption is minimized. Because what happens is one of the main ways that antibiotics can really disrupt the microbiome is that they bring down the microbial load quite significantly. So a study being done at Royal Holloway London University showing that a single dose of Augmentin, we're talking about 650 milligrams of Augmentin, reduces the microbiome count by 90%. So one dose knocks down 90% of the microbes in the microbiome, right? Now, they bounce right back. But what bounces back at what levels is where you get the perturbations. So what happens is, when you knock down the microbiome load by 90%, all of those lactic acid producing bacteria are now suppressed for a period of time, which means that the bowels start to become more alkaline. And when the bowels are becoming more alkaline, then the species that do well in an alkaline bowel, like Clostridia, tend to start to grow better, like E. coli, Clostridia, Enterobacter. They do better in a slightly more alkaline environment, and so they start to grow at a higher rate when the microbiome is bouncing back. So one of the ways of preventing that is that you take microbes that are stable in the presence of antibiotics, like Saccharomyces boulardii, that produces a lot of lactic acid in the gut. Even Pediococcus can do that, or the spores in Megaspore, they produce L-positive lactic acid. So during the course of antibiotics, they keep the gut acidified. So as the microbiome is bouncing back, these non-acid favoring disruptive microbes don't come back at a much higher rate.
Gotcha. Another question that a lot of people ask us is, what's your favorite lab to check the microbiome, and how do you feel about it?
Yes. Oh, the microbiome test. So I, I don't right now, we don't use any sort of gut biome testing. It's not quite accurate, at least down to the species level. You can get some idea at the genus level, or even higher than that. But at the species level, to try to figure out what's wrong with a person by doing a stool test is a really daunting task. And the reason for that is the stool test is incredibly inaccurate for a few different reasons. One is, many of the companies that do the stool test, they'll use 16S ribosomal RNA testing. 16S ribosomal RNA testing simply does not have the resolution to get down to the species level. Also, when you run the sequencing, most people use Illumina sequencing. The way you hyperplex the probes will depend on what your chances are of picking up the DNA or RNA fragments in the bacteria. So even though they're there, your probes may not pick them up. And so there's a lot of laboratory technique that goes into that. Even when they do pick them up, what you get out of the sequencer is a whole mess of this raw data. All that raw data has to be translated through a bioinformatics pathway or a software, and each company seems to have their own bioinformatic software. So how that data is interpreted can change from company to company. So what we found in the clinical studies that we've done, because we wanted to add stool testing in the leaky gut trial that's publishing on August 15th, which we're happy to announce on this. I'll talk a little bit more about that. But what we want to do is add a stool testing component, but we knew inherently that stool testing was inaccurate. So we tested this. What we did is we took 5 people and we gave each of them a stool test every day for 5 days, right? So if there was a certain degree of accuracy within the stool test, we should be able to take the data that comes out without the names on it and match the 5 tests to each individual person. There should be some consistency within their 5 tests in 5 days in a row. What we did is we sent all that data to a biostatistician and we asked him to match up the tests with the individual. And he wasn't able to do it because it was all over the place. The data on day 1 versus day 2 versus day 3 was kind of all over the place. There were no real accuracies to it. So you can use the stool testing as like part of a clue, but I really wouldn't count on it because I've met so many practitioners who are hitting their patients with strong antimicrobials and things like that because a single stool test showed an overgrowth of some species of bacteria when that could be a false positive or an inaccurate count of that particular microbe. It's not a great way to go about it. If you're going to do it, I would send it to the American Gut Project rather than any of the labs, and then actually just look at it from a genus standpoint to look at the diversity rather than looking at species. That was a very long answer, but very important, very, very important subject because
People
so many people
appreciate
use it.
how thorough you are, so that's good.
I hope so. Or they're just all tuned out
Yeah, no, there's
10
so
minutes
many
into it.
questions. We're gonna take about 10, maybe 15 more minutes. We'll try to knock out some of these questions for you guys. I know you're all very eager to hear Karan's answers.
Well,
Okay.
and one of the things maybe what we can do, Acacia, for questions we don't get answered during the time,
Yeah,
we'll send out an email with answers to.
yeah, because I can find all your email addresses from your questions, so don't fear. Okay, the next question is how and when would you address parasites in this context? Because I know we touched on it briefly.
Right. So parasitic chronic infections are really interesting because we're starting to find out more now that parasites actually have a very important function within the microbiome and the immune system. In fact, there are studies that show that low parasite levels are associated with really high immune dysfunctions. So it's really hard to pinpoint right now if something that the person is suffering from is only due to a parasite. Um, the other part of it is parasites are very hard to control by external methods, meaning adding in an anti-parasitic or something like that, some compound that you're trying to use to target the parasite. The best way to control parasitic infections or overgrowths is by increasing the commensal bacteria because they tend to outcompete the parasites and keep them in check. Also, upregulating your own immune system, in particular secretory IgA, because your secretory IgA has up to 12 months of memory. So at any given time, you've got immunoglobulins within secretory IgA that remembers what a parasite looks like that exists within your body and can neutralize it. And when it neutralizes it, it helps the body get rid of the parasite. So one of the best ways to disrupt parasite is, again, to increase the microbial diversity, increase secretory IgA production. It's a little bit harder to go after the parasite specifically with antiparasitic treatments.
Okay. We have a C. diff question for you. There was a question about would a low-grade C. diff infection show up on a test?
If you're looking at a stool test, yes, it can. So C. diff can show up at any time, even if you're not infected, because C. diff is a normal part of the commensal flora. So what they're looking at is certain levels of C. diff. And some tests are even looking at metabolites or the toxins specifically. So C. diff has a couple of characteristic toxins that it produces. That's really much more of a accurate test to see whether C. diff is an active ongoing infection right now in your gut. So low levels still can show up. And it's not surprising to think that a lot of people will have some form of low-level C. diff infection going on in an acute part of in a specific part of the bowel, not necessarily systemically. But C. diff, again, is opportunistic. So it only grows when there's perturbations in the microbiome in that part. It's not as virulent as people think. So simple, adding in like the spores, for example, can control that. But yes, you can get detection of low-grade C. diff infection.
Okay. The second part of that question would be, for C. diff, would you use Megaspore or Restore Flora, or would you use them together?
So great question. We've actually been experimenting with this quite a bit in our clinic, and we have some really good case trials. What we do is we use three different probiotics. We use Megaspore, we use Restore Flora, and we use the high-dose HU58. And the way we use it is we do two or three Megaspore caps a day. We do 2 caps of the Restore Flora. We do one in the daytime and one in the evening, and then we do one, and sometimes depending on how severe this case is, 2 caps of the HU58. The HU58 can be taken morning or evening with the Megaspore dose or outside of the Megaspore dose. The combination of those three seems to do a really good job at controlling C. diff and actually making it almost undetectable a few weeks after treatment. What we find, though, is that once it's undetectable, if you immediately stop the other two and just stay on Megaspore, you may get a recurrence. So we keep those patients on that regimen for a couple of months before we start tapering down and going to just your Megaspore maintenance with 1 or 2 Restore Flora, and then a couple months down the road, we just remove the Restore Flora as well. But acutely, the combination of the three seems to be really powerful so far, and we're actually working on some clinical trials to look at studying those more formally.
Awesome. Lori asks if Megaspore is effective for acute food poisoning.
Yes, actually, and that's, I think, it happened to me and family members when traveling, for example, to Thailand. We had an acute case of it was definitely some form of food poisoning. It seemed to have about a 5 or 6 hour incubation period, which is typically normal for certain types of food poisoning. It was associated with projectile vomiting, chronic diarrhea. This wasn't on me, or a family member, and this was in Paradise in Thailand, and so it was not a good time to have it, and it could have been it was associated with something we ate that morning. I dosed them up in Megaspore. She was taking about 5 caps twice a day. So the first time the symptoms came on, took 5 caps, as much as it could hold it down with water. Of course, a lot of that kind of came back up, so gave another 3 or 4 caps after that. One of the good things with Megaspore is that even though they're not going to be able to necessarily take the capsules and hold it down in the active stage of the infection, you can open up the capsules and mix it in water and take tiny, tiny sips of the water over a period of a couple hours, because they'll be able to hold in small sips. They're not going to be able to hold in 2, 3, 4 ounces of water at once. And so that's what they were doing. They emptied out 5 caps afterwards into a glass of water, sipping on that. About 4 or 5 hours after that, she was already feeling much better. So then we hit her with a little bit of food. So there was some carbon sources in there. It was just some simple bread, and then took another 3 or 4 caps of Megaspore. So about a total of about 8 or 9 caps in about a 8-hour period. The next day, she was 100% better. I mean, completely normal, no symptoms at all. I've done that myself. I got to a conference one day, and there was something that I ate on the way or something. When I got there, I started feeling really sick. Had to run to the bathroom, wasn't pretty. And I took 5 caps of Megaspore, took a nap. I held it down, so that was good. And then when I woke up from the nap, I ate a little something, took another 5 caps, and 6, 7 hours later, I was 100%. So Megaspore can really help with acute gut infections. In fact, in Europe and all that, that's the prescription use for the spores.
Right. Okay, Sheila has a question about chronic UTIs. Do you have a protocol that you recommend for those?
Yeah, so with chronic UTIs, it becomes extremely important to address a couple of things. One is the health of the mucosa itself. So the gut mucosa reflects upon the urogenital mucosa. If the gut mucosa is not healthy, you're not producing enough secretory IgA. Your urogenital mucosa will not have adequate amounts of secretory IgA to control the overgrowths of some of the other commensals. Now, what's interesting about the urogenital tract is that diversity of the microbiome tends to be really low. That's one of the couple of places in the body where diversity should be low. You really have 5 Lactobacillus species that are the predominant species. There's a few other species that tend to be really low levels, but if those 5 species are not predominant and you start getting overgrowths of other species, you start to increase diversity, you'll start to develop chronic UTIs and other issues, chronic BV and so on. So it becomes important to have those 5 species. Now, what's interesting is that the secretory IgA within the urogenital tract seems to know what species should be there, not be there. So if you're producing enough IgA in the mucosa, it tends to neutralize the species that shouldn't be growing, allowing for the crispactus and reuteri and all that that should be there to start to grow. So the important thing would be to fix the gut mucosa. And we talked about ways of fixing the gut mucosa and increasing secretory IgA. We also have many practitioners that use a single cap of Megaspore as a vaginal suppository. So you can actually, at night before bed, just insert the capsule itself, or remove it, mix the contents in some sort of cream, and insert it. But the capsule seems to work just fine. It dissolves well. And then, the use of cranberry and all that can be useful if there is an acute infection going on. So cranberry extract can help dislodge some of the microbes and prevent them from actually causing infection. But the way to prevent that chronic UTI from coming back is going to be fixing that mucosal system.
Awesome. Thank you. Can you speak to the accuracy of the SIBO breath tests?
Yes. So the what I call the exogenous breath test, the one that's done outside of the body, is mildly accurate. It's about 50-50. So you can get a false negative, and that's one of the problems with it. Typically, you won't see a false positive, but you can get false negatives. I've had numerous people that will say, "Oh, but I was negative on the SIBO breath test," but then when you do the aspiration test, which is one where they go inside, that's a lot more accurate. Then it shows actual positivity. Now, SIBO, though, is almost it's easier to detect physiologically without even doing a test. So because it's a small intestinal bacterial overgrowth, you can just test the patient by giving them something that's fermentable, like a single glucose monomer or disaccharide, something that has glucose, fructose in it that we know will exaggerate SIBO symptoms. And then if they start experiencing the symptoms within in about 2 hours after taking it, or within about 90 minutes after taking it, then it's more than likely SIBO. So if they're getting that bloating in the bowels, anywhere from 90 to 120 minutes after taking foods that tend to be high in glucose or fructose, then they more than likely have SIBO. Now, one thing that I've seen that people mistake for SIBO all the time is intestinal inflammation. So people will tell me, "Oh, I have really bad SIBO," and I'll say, "Okay, well, how much time after eating do you get the bloating?" They go, "Oh, everything makes me bloat. Even drinking water makes me bloat." And I'm like, "Well, that's not SIBO, because with SIBO, water shouldn't necessarily make you bloat. That's intestinal inflammation." So inflammation within your intestinal lumen itself can cause the intestinal tract to actually swell, which pushes everything out and makes you feel and look completely bloated. That's not necessarily SIBO. That's intestinal inflammation because of early onset things like colitis or gastritis. It's your intestinal epithelial cells that are totally irritated and inflamed, and almost anything you put into your system can cause that inflammation. And I think we talked about a friend of yours, Acacia, that may have that kind of issue where she's just always bloated and there's always distension in the belly, both to the side and forward. And so a lot of times when you see it both to the side and forward, and almost anything can irritate it, it might not even be SIBO. I mean, SIBO may be part of it, but you've got something bigger there. More likely something that's more like a enteritis.
Right. Yeah. Okay, we have another question about how megaspore would affect a hepatitis infection from a needle stick.
Sorry, say that again.
The question is, how would megaspore affect a hepatitis infection from a needle stick?
That's a good question. Megaspore is going to always improve the function of your immune system, whether it's the local immune system in the mucosa. We know that it reduces inflammation in the mucosa. We also know that it can upregulate peripheral immune function, like peripheral macrophage cells that need maturation that is stimulated by the production of short-chain fatty acids in the gut. So your peripheral immune system is dependent on the health of your gut. I mean, can it prevent the onset of hepatitis? Probably not. That's especially from a needle stick where it's going right into the blood. But can it help prevent the long-term impact of hepatitis, which is on the liver? And absolutely, that's it can be hepatitis can be controlled through improved immune function. Encephalopathy study on the spores, where these are liver failure patients that have numerous other comorbidities that come from increased ammonia levels. And we used the spores on them. They tolerated it very well. It brought down their blood ammonia levels and dramatically improved their lifestyles as well, and just the quality of life. We got a lot of great quality of life report. That's a study that's being written up for publication. So it can help, but it's not going to prevent the onset of disease once the needle's stuck.
Right. Okay, a couple more, then we'll wrap things up. Okay, the next question is, do you have any comment on megaspore and the role that it would play in mast cell disorders?
No, I've actually wrote an email to somebody about that. We don't have any direct correlation with, at least we're not testing it right now in mast cell disorders. I would say, however, that mast cell disorders could be linked to chronic infections. So there is a link here in excessive production of histamine, IgE, and things like that present themselves as allergic type of reactions that are triggered by chronic infections. So histamine intolerance, for example, there's a nice tie to chronic viral infections and histamine intolerance, because one of the ways that your body signals to your immune cells that there's an infection is by producing histamine. Your microbes produce histamine. Your microbes within the microbiome. So histamine and taurine are used as these chemical signatures. And so there seems to be some tie. It's not very solid yet to chronic infections, but I think most immune dysfunctions in general can be tied back to some degree to a chronic infection. So in that respect, I think megaspore can help, but I can't say that we've got a clinical study or case trial where we treated mast cell dysfunction effectively with it.
Right. Okay. So as the last question, there have been a number of questions along the lines of, can we treat gut infections or the dysbiosis just via diet? Why do we need to use supplementation? So can you speak to that?
Yeah, so and you can start to improve it via diet, of course. But one of the problems is compliance within your patients, right? Your life would be so easy if you could just tell your patients, okay, starting from this afternoon, these are the things you eat and these are the things you totally avoid. But the number of your patients that are actually going to follow that to a T are going to be very, very small. And small changes in their diet, maybe they're eating really well and then they all of a sudden have a cupcake, and that's enough to cause more disruption changes. So you kind of have to give them an edge. That's where the reconditioning microbes like the spores come in really handy, because they allow for the gut to become reconditioned the way they naturally would anyway. And then any positive changes in diet will be amplified, and any bad decisions in diet can be dampened because the spores are in there kind of take care of some of the perturbations that would occur from them not following a diet. Diet is going to be extremely important. If they're not following some sort of diet to try to help the infection, they're never going to, no matter what they're supplementing with, they're never going to recover. But it's going to be really hard to do it with diet alone, only because or at least mainly because of the compliance issue with patients, I would say. Also, it becomes really hard for patients to eat a diverse enough diet, complex enough diet to allow their microbiome to naturally change. But again, nature has given us these natural organisms that are abundant in the natural environment to go in and do some of that fixing work for us. So that's why we think it's so important to add those microbes, the spores, along with the dietary changes.
Yeah, definitely. Awesome. Okay. Well, I think we're going to, I know there's a lot more questions left, but we will address them all afterwards so that Karan can go to bed and sleep, because that's really important
Yes,
for all of us,
it was
right?
on the slide. Absolutely.
Probably all very tired. But if you guys will tune in next time, next month on September, we'll announce the day. I believe it's like the 18th or something. We're having our next episode on the gut-brain connection. So we'll be talking all about that. And Karan will have another presentation. We will send out the slides from this. So if you came in late, you can still get those. We have all your email addresses. If you have questions for us, let us know. But thank you so much for being with us.
But
and
you can pull the questions that we didn't answer, right?
Yeah.
We could send as a mass email the answers to everybody so everyone sees the answers.
It'll be like a fun chat room.
Yeah. Well, thank you, Acacia. So nice to see you.
Good to see you too. All right.
Thank you all for joining
Good night,
us.
everyone. Bye.
Bye-bye.